Could something as simple as switching from white bread to whole grain bread save your vision as you age? That sounds almost too good to be true, but that's exactly what Dr. Alan Taylor's 4 decades of research at Tufts University suggests. Age-related macular degeneration is a leading cause of vision loss, and most of us are unknowingly accelerating it with our diet. Dr. Taylor has discovered that high glycemic foods such as sodas, processed foods, basically anything that spikes your blood sugar, create what he calls a double jeopardy in your eyes. You're creating damaged proteins while simultaneously losing your ability to clear them out. However, he's shown in mouse models that switching from a high to low glycemic diet can actually arrest AMD progression. So the practical steps are choosing oatmeal over sugary cereals or whole grain over white bread. Glucose spikes are never good, and this same dietary change protects your heart, your brain, and potentially reduces cancer risk. One simple change, multiple systems protected. Dr. Taylor breaks down exactly how this works, what the early warning signs are, and what you can do starting today. So let's dive into it. Hello, Dr. Taylor. You have run the Laboratory for Nutrition and Vision Research at Tufts University for 4 decades, pioneering groundbreaking discoveries on how a diet and nutrition can prevent or even reverse age-related diseases like macular degeneration, cataracts. So welcome to Modern Healthspan, and thank you so much for joining us today.
Nice to be with you, Richard.
So, Dr. Taylor, your research is kind of the intersection of nutrition and eye health, which is quite a unique combination. So can you give some background as to what led you into those particular areas?
Well, I actually began my career as a chemist, then switched to biochemistry. And as a biochemist, my first challenges were to isolate enzymes, which had to do with degrading proteins. And I isolated the enzyme initially from the lenses of cow eyes. So I single-handedly cut apart 20,000 cow eyes to take out their lenses. And the lens, as you remember, right, is a clear organ. It helps to focus light on the retina. And in such, it's sort of like a diamond, a biological diamond with such beautiful light transmission capacities. And I isolated this enzyme, then we did a lot of protein chemistry on it, learned how it works, its mechanisms and biochemical functions. And all that time, I never looked behind the lens, right? The lens is interesting in that it's made of a single cell type. So isolating proteins from it is relatively easy as long as you can get the lens. But what does the lens do? The light comes in through the cornea, passes on through the lens, and then gets focused on the retina. If you look back at the retina, now if you put your fingers together, your thumb and your forefinger, the retinal lens are about as far one from the other as you can bring your fingers without the two fingers touching each other. So you have this lens, which is a very simple cell system, and it focuses light on the retina, which probably has 200 or 300 different kinds of cells in it, right in back of it. So the retina has a much more complex job. It doesn't just focus the light, but it also has to change the light into a chemical and then electrical signal, right? So it has to have all kinds of different kinds of cells. And after working on the lens for about 20 years, we became interested in the retina because even to date, there's no cure for age-related macular degeneration. And when the light is focused on the retina, it's focused primarily on an area of the retina called the macula, and that's the zone of high resolution vision. So we began asking questions about the retina, and we asked, what's comparable between the lens and the retina? The lens is actually made largely of cells that are pretty much dead. They throw away their nucleus. The retina, all the cells, of course, are nucleated. And whereas the lens exists without any blood supply, the retina has two blood supplies. So the lens is almost like a moribund tissue, just functioning year after year, decade after decade, transmitting light until a person gets cataracts. And as compared to the retina, which is a very dynamic tissue, it probably is the most rapidly metabolizing tissue in the body. And the photoreceptors that receive the light are actually renewed 10% every day. So every morning when you wake up, you get 10% new photoreceptors. It's like a push-up ice cream cone that every day the photoreceptor pushes out, it's out at 10%. That gets eaten up by the resident retinal pigmented epithelial cells. And so that you keep getting refreshed photoreceptors, which just from a teleological point of view, that seems logical because there's so much energy going into transforming light to chemical to electrical energy. You might think, well, there's a lot of oxidative stress there, a lot of burnout. So of course, let's replace those tissues as often as we can. So I got very interested, asking, what's the difference between these two tissues, which exist really within millimeters of one another, and how are they metabolically so different? And might there be some common phenomena that may happen in one tissue that also happen in the other tissue? What can we learn from one to the other? And then it turned out that maybe we could apply what we've learned in one cell system to the other and really preserve people's vision. And then that led us on to a whole new trail of what kind of questions we ask, what, for instance, intakes, what food intakes, what environmental phenomena might affect the fidelity of the operation of these two tissues, the lens and the retina, and now primarily the retina. So I hope I've given you a little bit of introduction, and I could specify particularly what the mechanisms are that we are focusing on.
Absolutely want to dive into that. So you talked a bit about how the lens changes with age. I mean, it basically doesn't. It just gets cataracts. It was what I understood. But how does the retina, how does the retinal cells, how do they change as we get older?
Well, so cutting to the chase, to the very bottom line, is you lose some retinal cells, right? You start to lose photoreceptors. The photoreceptors are the cells that receive the light signal, the chemical signal that becomes electrical signal that gets fed onto the brain, right? So through a variety of other neurons. But so you start to lose those cells. So, and that results in areas of blindness, which we call like geographic atrophy. Geographic atrophy means there's certain cells in certain parts of the retina that start to fall away. And that's one stage of age-related macular degeneration. The other form of macular, and that's called sort of dry macular degeneration. The form that's called wet macular degeneration is you start to bleed. The very, very fine blood vessels start to leak, and blood gets into the retina area, and it starts to separate the photoreceptors from the retinal pigmented epithelial cells, which are the cells that have to ingest all the shed photoreceptors every night. Well, so in short, I've told you the two
Yeah.
ways that the retina changes with age. It starts to either lose photoreceptors due to geographic atrophy, which is called dry AMD, age-related macular degeneration, or wet age-related macular degeneration, where the blood vessels break and they start to leak blood into the photoreceptor area. Well, that's the short answer. Now, the slightly longer answer is, so what? What does that blood do? Why is it a problem? Oh, what it starts to do is it starts to separate the photoreceptors from the retinal pigmented epithelial cells. Now, in order to continue to function, you have to have a way to get rid of the debris that the photoreceptors are shedding every night. And that gets shed into photoreceptors, into the retinal pigmented epithelial cells. So if you lose that connection, a lot of debris starts to accumulate, and the retinal pigmented epithelial cells start to die as well. So you start to basically have a plumbing system where the outflow is blocked. And because the outflow is blocked, the cells above it, all the pipes coming into it, all get backed up as well. And so basically, you have a disconnect in the transmission system that should be bringing electrical signals to the brain.
So it sounds from your description that AMD, like dry and wet, are significantly different. Is that correct? Because it's like it says age-related macular degeneration. It sounds like it's a single disease, but the dry and the wet seem to have very different causes.
They do have two different etiologies, but they are related in that usually dry AMD is a precursor to wet AMD. One tends to follow the other. So if you could stop AMD at the dry stage, then you have a chance of preserving vision.
Mm.
And we will get into that in a little while because that's sort of the focus of our research. Because when you have dry AMD, you still have a lot of retinal function retained and maintained. And you can live very happily with a loss of a lot of your photoreceptor cells. But at a certain point, that you pass a threshold where blindness ensues. So our objective is to try to maintain as much of the retina as possible in as early stage of degeneration as possible, so that you don't progress to the point where you lose a lot of visual function.
Right. And your research, I think, in particular, is looking at the nutritional impact on AMD and the causes. So what part of the nutrition is the danger for AMD?
Okay, I think maybe a question I might want to ask is what part of nutrition is useful, to preserve, retinal function. And so there have been intervention trials called the AREDS trials, which were done 2 decades ago now. The age-related eye disease study, that's AREDS, the acronym is AREDS. And they found that for people with early stage AMD, and I'm talking about sort of very early dry AMD, if they take or have sufficient supplies of combination of vitamin C, vitamin E, a little zinc, some lutein, and that seems to be associated with preservation of vision. And in terms of food, specifically eating fish, right? Oily fish is very healthful. Now, you might have heard the same recipe before because cardiovascular disease responds very similarly. So you get a double benefit for consuming these kind of diets. Now, you might also want to ask, okay, if I eat that diet, what am I not eating? Because if I'm eating certain things, I'm probably not eating other things. And that's sort of what clued us in a way, is what are you not eating? If you're eating, quote unquote, the Mediterranean diet, or something like the Mediterranean diet, you're probably not consuming all the sugar, sweetened beverages, and all the high glycemic index foods that are in the Western diet. Like our diet, the Western diet in America, is characterized by consumption of an awful lot of sugar sweetened foods, right? I think of something like 80% of the foods on the shelves in the supermarket are sweetened in some form or another. And the typical American, I think the statistic is something like drinks about 50 gallons of soda a year. Now you might say, wow, 50 gallons of soda, that's so much soda. Now, who drinks that? Well, if you break it down to a day, it's really only 1.5 cans of Coke or something like that. So it does all of a sudden something that seems relatively mild in the aggregate is an enormous amount of soda. And why am I focusing on soda? Because sodas contain an enormous amount of sugar. You've got probably over 8 teaspoons of sugar in a single can. So if we're not eating that food when we're eating the healthy diet, what's the problem there? What's in that soda that might be damaging? And we start to focus on the sugar. And we ask if in people who consume a high glycemic index diet, like typical American diet, and we compare them to people who consume a low glycemic index diet. Epidemiologically, we observe in every cohort, whether it's Australia, 2 cohorts in Australia, a cohort here, and cohorts in Europe, every single cohort suggested that consuming the high glycemic index diet puts you at considerable greater risk for age-related macular degeneration, even if you're not diabetic. And that's crucial. That's even in normal people who are not diabetic. And in fact, the risk for developing age-related macular degeneration is almost comparable to the risk that you incur for smokers or for genetic predispositions. So the risk of increased age-related macular degeneration is very significantly associated with consuming a high glycemic index diet. Now, I probably should define what a high glycemic index diet is, or do you know that already?
Please explain.
Um, so a high glycemic index diet is defined by the amount of sugar that it delivers to the blood. So in other words, if I have two foods, one of which is made of, say, a whole grain cereal, and say it contains 10 milligrams or 10 grams of sugar, and I have, say, a candy bar, which also contains 10 grams of sugar, and I eat the two of those, and I look at my blood, say, within 30 minutes, I find that if I eat the candy bar, my sugar level spikes, goes up very, very rapidly. Even though the candy bar has the same amount of sugar as the whole grain food, the whole grain food doesn't result in such rapid accumulation of blood in the sugar, of sugar in the blood. So the glycemic index basically rates one food versus another food. So the standard food is 50 grams of sugar, and we compare the amount of sugar that's delivered to the blood in various other foods. Could be an apple, could be a pear, could be a potato, could be potato chips. Um, and so foods that are very high glycemic index have 100%. They deliver the same amount of sugar, say in 30 minutes, as actual sugar per se. And low glycemic index foods like whole grains deliver the same amount of sugar, but very gradually. So the secret here, we think, is that the cells never see the blast of sugar. Cells are able to handle low levels of sugar coming into them over a prolonged period of time. But we think that they sustain significant biochemical damage and then functional damage when they see the high levels of sugar. Now, moving on to a little bit about the mechanism, we think, that in order for a cell to function, it has to degrade the damaged materials and get rid of them, secrete them. And there's a whole mechanism for that in the retina. One of the blood systems in the retina, his chore is to deliver nutrients on the one hand, and on the other to take out waste materials. And that happens through the retinal pigmented epithelial cells, which I mentioned earlier. So if these retinal pigmented epithelial cells, instead of degrading the materials, they accumulate the materials, basically they're blocking up the disposal system. It's like having a blocked toilet. And we think that the sugar is damaging both the enzymes that are responsible for degrading damaged materials, like protein degrading enzymes called proteases or nucleases, enzymes that might degrade genetic material. And the enzymes themselves are damaged. So the proteins per se are even damaged. So basically, the sugar is damaging everything that ought to be part of normal metabolism and normal editing of the cells, and instead is resulting in sort of the corruption of these processes. And therefore, the cells cease functioning properly, and therefore the organ stops to function properly. I hope I've said that in a way that's intelligible.
Yes, yes, absolutely. So would a ketogenic diet be kind of better? I mean, like no sugar at all?
Well, it depends but then what are you doing with the ketones, with all those ketones? The ketones are not necessarily very helpful either. So I am not a fan of extreme diets, but that's my personal bias. I think the secret to to the best metabolism, you know, is obviously having good genes, and then you select your parents carefully, and the other is to probably just eat less of things. I mean, Americans are terribly overweight. A third of us are obese. I think consuming, I would say, a Mediterranean diet, which is rich in fruits, vegetables, and I'd add fish, as compared to our diets, which are too rich in, sweetened foods, processed foods. I think that really would deliver the best health benefit because it also gives you high levels of the nutrients we talked about before, right? The vitamins and cofactors. So I think staying on a rational diet and not eating too much of it is probably as good as a special ketone diet. So the secret, of course, in that therein lies in choice and self-control, which people don't like to hear. But I think there's a lot of benefit to be gained from it.
Yes. Well, the one thing is, different foods spike sugar in different people. I mean, I understand that it's not uniform. And so you would need to kind of research what spikes sugar in you.
Yeah, that's true. But I think there are many generalizations that are valid. And I think those could rule the day. For instance, if you drink a can of Coke, no matter who you are, my bet is you will spike. The sugar level will spike. My bet is if you eat a piece of cake, whatever kind of cake you like, um, my bet is most of us will spike. My bet is if you drink a soda, every day, you will spike. And by the way, what I didn't mention before, and which it's useful to think about, is that we now have the capacity to integrate the glycemic index of every food with respect to other, in the whole diet. So we can talk about a low glycemic index diet as opposed to just a low glycemic index food. And it's true that some foods affect the metabolism and absorption of others. And of course, all these affect your microbiome as well. So each of us has a slightly different microbiome. And all those do affect um, our absorption and metabolism. But I think in general, one can assume that if we eat these very sweetened foods and a lot of them, you are putting your body under a lot of burden that it doesn't need to be under. Um,
Right.
so it's true. I mean, I know I wore as a test a continuous glucose monitor just to see what my dietary responses are to foods. And they were pretty predictable. I probably could have read a book and seen comparable responses as to what I did show on the continuous glucose monitor. On the other hand, I have friends who register slightly differently, but they're not major differences.
Right.
I mean,
Yes.
remember, we're integrating information. Our bodies are integrating information not over minutes and hours, but they're integrating. I mean, we are the sum of all of our parts and the parts that went into us over years. So I think maybe one might respond differently one moment to moment from another person. But overall, I think, at least from a statistical point of view, most of us will show comparable responses to dietary burdens.
You mentioned that you could think about a low glycemic diet rather than a low glycemic food. Is there a resource that would help us to determine a integrated glycemic index for like a plate of food?
Yes, I think if you look online, you can find a variety of resources that talk about the glycemic index of various diets and foods. I think this has been done by several studies. Most of those of us in the field, I know we've done it. We haven't published that list, but there are comparable databases available. And certainly people in Europe have also published databases like that.
If you are, so if you, so we talked about these spikes, and the spikes have this bad effect. If you have generally high blood sugar, so you're like pre-diabetic, does that have a significant increase in the risk of AMD?
Well, it's a very important question, and people have been asking this question for years. And a problem is that diabetics get diabetic retinopathy, and their blood vessels are very weak, so they tend to get disease a little bit earlier. So sometimes it may obscure the relationship between age-related macular degeneration and other retinal diseases. So it depends on who you speak with. I think the data tends to suggest that there is a correlation between pre-diabetic conditions and susceptibility for age-related macular degeneration. That once you start to cross that threshold and move into diabetes, that becomes a little murkier, but there's still an association. But some studies say no, and some studies say yes. But the problem is, it's not that the diabetics don't get age-related macular degeneration and they're scot-free and their retinas function fine. They just get diabetic retinopathy, which is even worse.
Leaving AMD for a second, the other thing that you mentioned was cataracts. So we get cataracts in the lens. So could you talk about what exactly are cataracts, and is there anything we can do about them?
Let me first describe what are cataracts. So the lens is made, as I said earlier, of a single cell type. That cell goes through various stages of what we call differentiation. But if it goes from a little round thing, like a little round ball, say, and it becomes like a fiber optic. So the lens, if you cut the lens apart, the lens used to be called the crystalline lens. And why crystalline lens? Because when you have a crystal, it has a specific shape and structure, a crystal of a rock, for instance. Like you hit the rock and it fractures along a specific line. And actually, the same thing happens in the lens. If you prick the lens with a needle, it also fractures across a certain line. That's why the analogy is made to crystals. They call the crystalline lens. And what are those crystals? Those crystals, really, I'm going to now change the term to fiber optic, because nowadays we know what a fiber optic is, right? It's a cable that conducts light. And so the lens is basically like if you cut it, or like rings of a tree, if you cut it across, it looks like rings of a tree, all made of a single type of cell. And those cells are full of proteins. Those proteins are called crystallins, again, from the analogy of crystalline lens. And there are a very limited set of proteins, maybe a dozen proteins that make up the lens. And what happens as those proteins get old, instead of remaining in solution, a solution is clear by definition. A solution is something that's clear, like you dissolve sugar in water, it remains clear. But if you have too much sugar in the water, then you get crystals start to form, right? Every kid does that in grade school. You form sugar crystals. And so the same thing starts to happen with the proteins in the lens. They become damaged, not necessarily that they crystallize, but they just become damaged. So they no longer can function. And they also aggregate. So one protein starts to get attached to the other. And that's part of the problem with sugar. It acts like a cross-linker between proteins. And so the proteins become so big that they can no longer remain in solution, and they basically precipitate out, and they precipitate out within the cells of the lens. And as those start to grow, they obscure light because the light can no longer travel through the precipitated protein. And so that's really what defines a cataract. Then you get they just get larger and larger. The aggregates get larger and larger, so they no longer transmit light. And that that causes the opacification instead of the transmission of light in the younger cells in the younger lens. And that damage can be caused by all kinds of stresses. It could be caused by sugar. It could become by oxidation. It could be caused by the combination of sugar and the oxidation, which is what we think really happens. And then that's compounded by the fact that in the lens, there's very little opportunity for replacement, right? In many cells, you have replacement mechanisms. You have genes that encode new proteins. But in the lens cells throw away their nucleus, and that's part of the way they develop total clarity. Um, and so they can no longer replace damaged material. So basically, everything gets old in a sort of concerted fashion, a coordinated fashion. But getting old like that, unfortunately, leads to more and more damage at a more and more rapid rate as the lens gets older, because you have less and less capability to remove the damaged materials. So as damage accumulates, there's no capacity to remove it. So the damage accumulates even faster. And that's why rates of cataract go up very extremely rapidly when we push, 70 years of age, 80 years of age.
Do we have any? Is there anything we can do about the cataracts? I mean, they sound kind of inevitable.
Well, we did years of research about that. We put in an awful lot of effort into it. And we think that we found diets that are protective, especially a diet that say high in vitamin C, but it's very easy in our culture to get high levels of vitamin C. Um, and we didn't find the same kind of robust correlations as we found with macular degeneration. And quite honestly, I'm not sure why. You might think that the lens would be a perfect place to find those correlations, but they have been a bit elusive. Um, something else I think was a little bit discouraging, although maybe it's a good thing, and that is there is an effective way to get rid of cataracts. So you go into the ophthalmologist, and half an hour later, you come out with a plastic lens. So it's not fun to have compromised vision, say between your 60 and 80, but you go to 80 and you go into the ophthalmologist, get a plastic lens. So there is a resolution for that situation. And we haven't done the same kind of intervention trial that we probably should do, because cataract still remains the major blinding disease of the elderly in the world. There's a lot of places where people can't go in to get to the ophthalmologist and get a plastic lens put in. So it remains a huge public health problem. And people are shunned because they're unable to see in parts of the world. But we still need to do the intervention trial that was done for age-related macular degeneration.
in in terms of trying to figure out what the best diet would be.
Yes, yes, but from an epidemiological perspective, we have a lot of data about that. And from an epidemiological perspective, that is not doing the randomized double-blinded control as was done for macular degeneration. From an epidemiological point of view, the data do suggest that consuming higher levels of antioxidants are protective against cataracts as well.
Can we
And I
do?
think we
Yeah,
probably have more than 20,000 people worth of data just from my lab alone. And that.
and the antioxidants you mentioned particularly were like, um, well, what are the antioxidants in particular that are good for the eyes?
Well, the ones that people focus on in general are vitamin C, right, ascorbic acid, and vitamin E to the extent to which it's an antioxidant, which is probably a mild antioxidant, but it seems either the foods that contain vitamin E or vitamin E per se seem to be quite healthful. And then maybe lutein as well. And then, as I mentioned before, it's what you're not eating too, and keeping the sugar low.
Right. We often see zeaxanthin with lutein. Is
Yeah.
zeaxanthin helpful?
Well, the data is not as rich about zeaxanthin as it is about lutein, for instance. I think lutein zeaxanthin often comes along with zeaxanthin often comes along with lutein, but I'm less secure about that information.
I believe there are eye drops for cataracts in like dogs in particular with carnitine. Have you did,
Yeah.
do you have any comment on whether
So
L-carnitine helps?
yeah, so there are these biological antioxidants like acetylcarnitine and glutathione, for instance. But getting them to the right place is non-trivial. It turns out that your eye is very well protected, right? You have a cornea, behind the cornea you have the aqueous humor, behind the aqueous humor you get the lens, behind the lens you have the vitreous humor, behind that you have the retina. So there's all these tissues, right, that something has to get through to get to the target spot, to get to the lens, for instance, which is pretty close to the front. But in front of the lens is the aqueous humor, and the aqueous humor is flushing relatively quickly. So, so whatever you put in an eye drop has to get through the cornea, and then from the cornea, somehow or other survive the flushing that's going on in the aqueous humor to then get to the lens and even then to get to the right part of the lens. So it turns out that it's a technological challenge to get these things in. And so I am unaware that eye drops at this point are preventative for cataract. And it sounds like a good idea. It's a good idea. It just, like many good ideas, I tell my wife I have great ideas all the time. It's just, they just happen to fail. You know,
Yeah.
you know.
Yes. Uh, so you mentioned omega-3 fatty acids while we were talking about, I think earlier about macular degeneration.
Yeah.
So they help. Do they help with just with AMD or with AMD?
Well, they seem to be associated. Higher levels of these omega fatty acids are associated with retinal health. I mean that has been demonstrated, and we found it also epidemiologically that's the case. With respect to AMD, I'm less sure about cataracts.
Right. If we're looking at kind of other nutrients like maybe zinc or the B vitamins, do you see any? Do you commonly see any nutritional deficiencies which cause bad eye health that people should watch for?
Well, it's a classic that vitamin A deficiency, of course, is associated with blindness, right? Because vitamin A is critically involved with the light reception process. Um, so that's a clear one. And it's been demonstrated that giving people, a small amount of vitamin A just when they're young preserves vision for decades. Um, but most people in most parts of the world now have sufficient levels of vitamin A. Still useful, especially in the underdeveloped countries, but certainly in the West, most more industrialized nations where food is pretty plentiful, we do have adequate supplies. Um, I'm less aware of vitamin B associations with eye health. So far, that has not come to the fore. But that's probably also because many people probably have sufficient levels. Um, and I'm not quite sure how that would play out. The biochemical mechanism, at least in my mind, I don't know. Obviously, I have plenty of areas that I could learn a lot about. Um, but it's not clear how the mechanism that would manifest itself. I mean, as long as you're having enough what we call reducing equivalents, that's the opposite of it. For oxidation, to reduce oxidation, you have reducing, right? Reducing equivalents, that's like glutathione or acetyl-CoA. And the thing is that if you start to, it's hard to get those compounds into cells directly. So you have to do it by some indirect mechanism. I mean, that's where the diet is very helpful because the diet enables you to deliver either the reducing moieties themselves or produces other things in your cells that then become those reducing compounds. And that's how you deliver the benefit. I hope I'm being clear.
Yes, yes. So I saw one thing in your research that you talked about AGEs, like advanced glycation endpoints, and they tend
End
to go up.
products.
End products.
Right.
End products. Uh, if you so you make them internally if you have too much sugar. You can also eat them if you
Right.
cook. If you cook sugary foods, I guess, at high temperatures. Are the AGEs from the diet incorporated into the body? Do we absorb them, and are they negative?
Yeah. So there's been a lot of attention devoted to that. And I think the findings are that whereas there are high levels of these advanced glycation end products in foods that are fried, for instance, toasts, um, but your body doesn't absorb a lot of them. So most of it never makes its way into the body. And so to the extent to which they add additional burden, so of course it's as well to keep them low, but they don't add that much burden because your body is making a lot of these advanced glycation end products itself anyway. And it's the dietary sugars that really are the culprit. More so, I would say, than the dietary advanced glycation end products per se. But it doesn't hurt to keep that low.
Right. Well, certainly
But I would
fried.
say that health benefit, the major health, if one had to choose a health benefit, the health benefit I would focus on would be limiting the amount of starting materials that produce bodily advanced glycation end products,
Yes.
like sugar. I think you can make an analogy like cholesterol, right? You can limit the amount of cholesterol containing foods you eat, but the real burden of cholesterol in the body is actually cholesterol that's made inside the body. That's why the statins are so effective, right? Because they limit the body's ability to make the cholesterol. So comparable situation with the sugars. I think, yes, you could limit the advanced glycation end products, and that's useful. But it's better even more effective to limit the extent to which the body is making them, the advanced glycation end products.
And the best way to do that is to keep the sugar low.
Right.
Yeah, in the blood. If we looked at presbyopia, does is that inevitable? Is there anything we can do about presbyopia? Is that affected by the diet?
Good question. And I don't think there's really good data about that as far as I'm aware. And maybe I may not be aware of the data, but I'm not aware that there is. But one could predict that there should be some relationship because the presbyopia is caused in part by the changing of the lens, right? And the extent to which you retain a pliable, youthful, quote unquote, youthful lens, um, and if to the extent to which you do that by consuming a diet that's, prevents the hardening of and the aggregation of the proteins, so you should have some preservation of vision with a healthier diet. Um, but honestly, I'm unaware of that data. I'll have to look it up myself. No.
Right. How would I tell what the health of my eye is like at any particular point? And at what age should I start worrying about this?
Well, I think probably around 40 people begin to have ophthalmological exams regularly. There you're thinking about presbyopia, thinking about, the lens starting to show some changes, but that's usually not very significant until say 60. And people begin to use glasses, right, at around 40, 45, quite often. So that's why I'm suggesting that age is the time to to have ophthalmological exams, so to retain, you know, high accuracy vision. Um, but the diseases don't manifest themselves like cataract, probably not till 55, 60. And age-related macular degeneration, similarly, maybe a little bit older, another 10 years older. But there are biomarkers. I mean, they're very clear biomarkers. And in terms of a cataract, the first exam, of course, they just look for opacification of the lens. That's easily seen with what's called the slit lamp examination. That's just minutes in an optometrist or an ophthalmologist chair. If you want to look at the retina, then there are two different ways to look at that, and the technology is changing rapidly. But one is to do what's called a retro exam and just look for what in the retina you see drusen, these little yellow spots. Now, the drusen is the first marker of age-related macular degeneration. Once the drusen start to accumulate and achieve a certain size, and we have a standardized size that we define as indicative of disease, that's the biomarker of beginnings of dry AMD, age-related macular degeneration. And as the drusen become larger and larger and accumulate more and more stable, they actually come and go initially, but then they stabilize and they become larger and they have a more diffuse appearance. That's more indicative of early age-related macular degeneration. So there are these biomarkers. Now, there's also another thing now called OCT, optical coherence tomography, which is a very rapid test that sort of scans your retina and gives a printout. So that's yet another way to examine the retina in the posterior portion of the eye. And I would say it's good to do that certainly after 60, because you want to catch disease as early as possible. And if it's going to encourage you to change your diet, at least to capitalize on whatever the dietary benefit can be, that's a good way to be alerted. And we're actually doing that kind of study. We're just starting a study this week in which we're starting to see if we can encourage people who have relatively early stages of macular degeneration to adopt a low glycemic index diet.
Is AMD reversible?
Well, so far, no. In a mouse model that we reared, it was either arrestable or reversible. We couldn't tell whether we just really arrested at an early stage or we actually reversed. I would tend to think that if you got it very, very early, and I'm not sure that clinicians can even pick it up that early, at this stage of technology. Maybe yes, but generally the best you could do is arrest it. But if you can arrest it, that's pretty good because you can function, excuse me, you can function with less than 50% of your photoreceptors intact. You can see
Right.
pretty well with that.
Yes, and so the dry AMD is that some of these photoreceptors are dying and not being replaced. So they're becoming senescent, or they're just dying.
Senescent and dysfunctional. I mean, you could be senescent and functional, right? Senescent just means you don't replicate. Well, photoreceptors are produced and they refresh themselves. It's not like one photoreceptor becomes another. They refresh themselves. If they become senescent and they don't refresh themselves, they become dysfunctional. Do you understand the distinction I'm making? In other words, a cell can continue to exist, right? Like say in a lens cell. A lens cell is pretty much almost by definition senescent because it throws away its nucleus and it lasts forever. And it might function very well for many, many years. But if a photoreceptor cell is senescent and it doesn't refresh itself, it will become dysfunctional.
Right.
So it might last for a while, but there's a good chance that it'll become dysfunctional. And
Okay.
so with dry AMD, you start to see like areas where the photoreceptors are either dead or lost, right? They just go away, and they don't have new photoreceptors replacing them.
Yes. So you lose some acuity in your vision because you have less, but you can lose a lot of these without really noticing it
Correct.
and functioning normally.
Correct.
And then hopefully by switching to a low glycemic diet, you can hold it there at least.
Correct. Yeah. And I think it's useful to remind people that this low glycemic index diet also benefits your heart, your circulatory system. So you're getting a lot of bang for your buck.
Yeah, absolutely. I mean, glucose spikes are not good for anything. I mean,
Right.
yeah, they're not good for your brain either. Well,
I
I
mean,
mean,
some people even would say that it's useful for preventing forms of cancer. And I think the data's not very strong yet, but seems like there's nothing lost. Oh, there's only gain.
okay. So what excites you most about the kind of current and future research for AMD? I mean, as you said, AMD at the moment is not curable. Well, at least we don't think it is. What do you see as kind of the next stage?
Well, to me, it would be very exciting if we can just exploit the data we have now for starters. In other words, think about can we really convince people with AMD to adopt a diet that will preserve their vision? And as I said, that study's beginning. Well, we're beginning a study to see if we could get them to adhere to a diet, to adhere to a low glycemic index diet. And if we can show that they do, then the next phase will be to put them on that diet and see if those who were put on the diet have delayed progression of age-related macular degeneration. That in itself would save millions of people from blindness. I mean, the ramifications of this are enormous. Um, I think we showed that if we predicted that if you decrease the glycemic index only 5 points, and I can tell you what that means, we could save 100,000 people from blinding AMD in a couple of years. That's an enormous contribution for such a mild change. And the change I'm talking about is changing the equivalent of only 5 slices of white bread. It's assuming your carbohydrate came from only 5 slices of white bread per day to 5 slices of whole grain bread. So that's a pretty mild change to achieve the change in glycemic index of the diet that would deliver the benefit. So that's one way to think about it. The other way to think about it is, okay, what about the people or maybe who physiologically unable to control the sugar? And we've tested multiple drugs in the lab already to see if in a mouse model we can prevent some of the retinal damage, and we can. So in non-diabetic animal. So this opens up a whole new opportunity for pharmacology. And then, of course, there's all kinds of new ways of increasing the protective capacities, either be it with pharmacologically or new kinds of drugs, or even one could think about genetic manipulations just to prove the point, even. And we've done that in the lab too. And again, we showed that if we could break down, like enhance the breakdown of these advanced glycation end products or preclude their formation, even in a high glycemic index context, we can diminish the effect of the damage to the retina, at least in a mouse model. So there's a lot of opportunity, I think, to diminish the ravages of age-related macular degeneration with up and coming, biochemistry, pharmacology, genetics.
Right. And sorry, it is the AGEs. So it's the AGEs that form inside the proteins that or attach to the proteins that
Right.
cause the downstream issues. Okay.
And by the way, we're also developing a whole new set of biomarkers. So the ages themselves, the AGEs, become biomarkers to the extent at which you can detect them in the blood. They augment the armamentarium of ways to diagnose disease. And we've published some of that. We published a lot of that already, even starting in 2017 or so. And so we're now figuring out, could we have a constellation, a group of AGs that tell you that in addition to maybe the heart being compromised, that the retina is compromised, even with a blood test. So that remains a possibility, which I hope to realize relatively soon, that we would have a blood test that would tell you your retina's susceptible to damage or at risk for damage.
Yes, that would be by looking at the AGE or the type of AGEs and the quantity of them in the blood.
Right. And the mix, the mix of age. Right.
Right. Okay, if listeners were to remember just one thing from this conversation, what would it be? Probably don't eat sugar.
Well, there's a lot of benefit to be gained by consuming switching to a low glycemic index diet, as opposed to the typical American high glycemic index diet.
Yeah. Okay, Dr. Taylor, if people want to follow your work, where should they go?
Well, we publish a decent amount in the scientific journals, in the ophthalmological journals, and we published a few lay articles or articles that are more oriented toward the lay public. You're asking me an important question, and I've tried to ask people to devote more time to converting our scientific data to something that's more lay approachable. Maybe they should look to people. Maybe they should look to people like you who translate hard science into something that's more approachable.
Yeah, that is good, and certainly, I mean, people can search on PubMed and find your articles. And yeah,
For sure. And
you
we
have,
will have. I hope that within 2 years from now, we'll have these results of the first study, first human, the beginnings of the intervention trial published as well. But that's a long time from now.
yes, but I think, I mean, your message is very clear, and if the thing is, you said, if you could persuade people to switch to a low glycemic diet who have AMD or have the startings of AMD, it seems to me like if I had that, I would switch, right? I mean, if you could convince me that it would stop me going blind, I would definitely drop my candy bars, no problem.
Well, I hope that that's the case. Ophthalmology, the people who review our grants, we actually put a grant in to do this work, to do the intervention trial, maybe 5 years ago or even longer than that. And the community was dubious, the community of judges was dubious. They said, look, can you stop people from smoking? Look how long it took. And so it might take a long time, but I would hope that with the possibility of retaining vision as opposed to losing vision, that would be a keen incentive. So I would hope it's sufficient incentive, but that's our first chore, is to demonstrate to the reviewers of our grants that people will be willing to adopt a healthy diet. I mean, why it's such a barrier eludes me, but I'm just one person.
Yeah, me too. But I mean, I guess it's like smoking. You have to get the message out and then just keep repeating
Yeah,
it until it becomes well known.
yeah, yeah. I think there's a lot of momentum now building with this with regard to respecting the benefits of consuming lower glycemic index diets. But I'd like to see that reflected in the physiology of the typical Americans. We'll see.
Yes. Yeah, we will. Okay, Dr. Taylor, thank you so much for joining us today. It's been wonderful talking to you.
I hope this is useful. And if you're able to read that, did you see the Progress of Retinal Eye Research review? I probably sent it to you. It's a very long review. It takes you right through from the fundamentals of how ages form to dietary applications. Um, that was an enormous review that I wrote maybe 3 years ago now, 2 years ago, and it's pretty up to date actually. Um, we have
Okay.
2 or 3 more sort of cutting edge articles coming out, but those will be a little while yet, more experimental. But if you haven't seen that, there's a journal called Progress in Retinal and Eye Research, and
Right.
if you look on the 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 last name of the first author was Bejarano, Begarano, Bejarano. But I could
Right.
send it to you if you don't have it. If you,
And I mean, if you could send me a link, that would be great.
yeah,
Um,
I can send it to you. Thanks a
Excellent.
lot.
Thank you,
Okay.
Dr. Taylor.
Good luck.